So thanks very much for the opportunity to speak at this meeting, and been listening in during the day, and it's absolutely fascinating. Martin Landry earlier in the day talked about clinical trials. So with the agreement of the organizers, I changed the topic because I thought he would, and he did, cover the topic of the clinical trials infrastructure. And instead, I wanted to discuss the opportunities that I think exist for Oxford researchers and clinicians to help UK Biobank to enhance it for dementia research. And I think hopefully the opportunities that would then provide for researchers here to take advantage of their local knowledge. So just to kind of set the scene in terms of UK Biobank and why studies like this are valuable. And as Cecilia mentioned, there are a number of large-scale prospective studies that have been established by Oxford investigators for which this is also true. The key advantage is that one can look at not just genetic determinants of health outcomes, but also environmental and lifestyle and the interactions between them. Because of course, in the kind of classical genetic studies that are done of case control, that's fine for genes where disease is unlikely to change the risk factor level, the genetics. It's not fine for those kinds of factors that may be influenced by disease or the prodrome to disease, and that is particularly important in dementia research. These resources, prospective cohorts, are particularly valuable when made available widely because they allow people to look at either the effects of a single exposure on many diseases, and the British Doctors study that Richard Doll set up with Austin Bradford Hill is a very nice example of that. It was initially set up to try to determine whether smoking was a cause of lung disease, of lung cancer. It then went on to show that it was the cause of many other kinds of cancers, chronic obstructive lung disease, vascular disease, etc. But it also allows the effects of many exposures on one disease to be studied. But the problem, if you like, is that only a relatively small proportion of the participants in such a study will develop any particular disease. And so in order to have enough individuals that develop that particular disease of interest, in this case, perhaps dementia, then you need to have large-scale studies and you need to have long-term follow-up. And that's particularly true, as I'll show you later, with respect to dementia. So what does big mean in this context? Well, Sarah Lewington in the Department of Population Health pulled together data from some 1 million people in 60 prospective cohorts from across the globe with about 10 years of follow-up. And then she randomly selected to demonstrate scale and the importance of scale, 5,000, then 50,000, then 500,000 of those individuals to look at the association between two strong risk factors, blood pressure and age, on the risk of coronary artery disease mortality plotted on the y-axis. So this is 5,000 of those individuals, and you can see very wide confidence intervals around the point estimates of risk plotted against blood pressure at different ages. Really very unclear picture. One goes to 50,000, then it's like a microscope, starts to come back into focus. But if you look at high blood pressure in older people, is there a flattening at the bottom end? Is there a J-shaped curve for younger individuals where lower blood pressure is associated with increased risk? Or as it all comes into focus, are these beautiful log linear associations whereby one can look at the interactions in relative terms? The slope is shallower at older ages, indicating a weaker association in relative terms at older ages, but it's a log linear plot. So the absolute differences at older ages are bigger, and so the absolute interaction is stronger at older ages than at younger ages. So it just demonstrates that even for common conditions like coronary artery disease mortality, strong risk factors, 500,000 people, it is just about enough to get clear pictures. So that was really the background to setting up some large-scale prospective studies. And at the beginning of the century, the MRC and the Wellcome Trust decided they were going to fund a study of 500,000 people, aged 40 to 69 at the time of recruitment in 2006-2010 from across England, Scotland, and Wales, located in different parts of the country to enhance heterogeneity of the cohort, and with very detailed information about lifestyle, environmental exposures, personal family history. Cognitive function was assessed using a battery that was designed by John Gallagher with colleagues, hearing and vision tests, and then some physical measurements and biological sample collection. And then consent from all the participants to access all of their medical and other health-related records. So not just health records, but other records that might be relevant to exposures that would be relevant to disease, and to recontact participants for further assessments, but that recontact would not involve feedback of information about them as individuals in terms of their risk factors. So if one looks at the kind of key dimensions that make, I think, UK Biobank uniquely valuable for research, the first one, the obvious one, is size, as I mentioned, but also it's the depth. I described very detailed information about lifestyle, environment, and medical history that has been obtained from the participants and also through linkage to their past medical history. But increasingly now that's being supplemented by an extensive range of biological assays as well as imaging. First off, and again, to illustrate Oxford's involvement in this, Peter Donnelly and others in the Wellcome Trust Centre for Human Genetics led a working group to work with Affymetrix to design a genotyping array of 850,000 variants with imputed measures for 90 million variants. And of note for dementia, many of these arrays at that time didn't have ApoE on, and they did ensure that it was on this array. The data from that genotyping was made available for 150,000 participants in 2015 and for the whole cohort in 2017. There have been GWASs of hundreds of traits done on these data, both looking at GWASs, genome-wide association studies for risk factors, be it height, weight, blood pressure, BMI, etc., or for diseases themselves. And this has been made possible because of its very large scale and the linkage into health records in the NHS. This is an analysis done by Genomics PLC, the spin-out that was set up by Peter Donnelly and the previous director of the Big Data Institute, Gil McVeigh, an Oxford spin-out called Genomics PLC. It was a PhD student actually at the Broad Institute in Boston who demonstrated, to many people's surprise, that you could combine large numbers of variants of small effect to identify a material proportion of the population who had risks equivalent to those with single gene disorders. So here at the top left, 3 to 5% or 3% of the population here that have a risk equivalent to BRCA1, BRCA2. Down at the bottom right, about 3% that have a risk equivalent to familial hypercholesterolemia. So these polygenic risk scores are now being piloted for their introduction into both preventive strategies and also into screening strategies for, say, breast cancer, colorectal cancer, etc. And there has been work done in dementia, looking at polygenic risk scores. Here are a couple of papers from July 2021 and from May 2022, looking at polygenic risk scores associated with Alzheimer's disease. Regeneron, a company based in Tarrytown in New Jersey, in New York, pulled together a consortium of funders initially with GSK, but then with other companies to fund the exome sequencing of the UK Biobank cohort, which was done within Regeneron's highly automated laboratories. the first 50,000 exomes for participants released here in Q1 2019, and then the full cohort have already been made available early in 2022. And an example of, again, work done in collaboration between Regeneron, UK Biobank, and Oxford. This is a combination of data from UK Biobank, the Mexico City Prospective study, which was set up by the Department of Population Health in collaboration with the Mexican Ministry of Health, and the Geisinger group in the US, identified a druggable target, a novel druggable target for obesity. You can see circled in the bottom left, of a rare variant associated with a substantial difference in body mass index. So these data, I think, open up the opportunity for identifying novel targets for a range of different diseases, focusing here on the exome, the 2 to 3% of the population, sorry, of the genome that actually produces the proteins. But of course, it's likely that the remainder of the genome is very relevant to the production of proteins, as well as the kind of the specific protein forming bit. And so it's important that we now have whole genome sequencing data available on the whole of the UK Biobank cohort, funded by the Medical Research Council and Wellcome Trust with 4 companies, with the Sanger and Decode in Iceland having done the sequencing, despite the pandemic, keeping much on track in terms of the targets for completing this work. And now Decode are running joint called analyses on the whole of these cohorts to make them more accessible to researchers. I think that's not necessarily the end of the sequence data work that will be done in UK Biobank. The Illumina platform that's used here with the short read sequencing does have issues around being able to assess structural variation, and also the ability to study epigenetics. Long read sequencing has been less accessible in the past, but I think it is now at the point where it would be operationally and financially feasible to do it at large scale, perhaps not quite cohort-wide for 500,000 people, but certainly for a large proportion of the UK Biobank population. And again, an Oxford focus to this might be around the work that has been done with Oxford Nanopore to develop their technology such that it could do large-scale long-read sequencing. And there are conversations going on to see whether that could be done, at least in a very substantial subset of UK Biobank as an initial step. But most of the participant biological sample is still unused. We have done hematological assays on the samples as they came in, various obvious biochemistry markers around lipids, glucose-related measures, and some cancer markers. But we're now starting to see other kinds of omic assays done. So Nightingale Health, a spin-out from a Finnish university, is at their expense doing their NMR metabolomic assay. Olink have been funded by another consortium led by Biogen to do proteomics. And the Chan Zuckerberg Initiative have funded a small pilot study with the Sanger, with Nicole Saranza, to look at RNA sequencing. Again, as occurred with Regeneron, this pharma proteomics project led by Biogen has added more and more companies, which has allowed them to go from the 1.5K Olink proteomic platform to the 3K proteomic platform. And those data are starting to emerge, and later this year will become available to other researchers beyond the consortium that's creating them. But we see these as pilots. We anticipate that the pharma proteomic project will move on to doing some platform, whether it's Olink or some other platform, on the whole cohort. And there are various other discussions going on around untargeted mass spec approaches. And we would very much welcome suggestions from the dementia community about what kinds of assays might be done. And it's worth bearing in mind that, for example, with the Olink proteomic platform, at the moment, they have total p-tau on, but they don't have the measurements that you would like, as I understand it from the dementia, but they could put those on. So I think the dementia community thinking about what it is they'd like to have on some of these proteomic platforms might be quite valuable, because it would be very straightforward for them to add them, as I understand it. And then if there is this initiative for pharma to fund the assays, then as a byproduct, those data would be there. Again, led by Oxford to a large extent, particularly the cardiac imaging and the brain imaging, a working group chaired by Paul Matthews, who was in Oxford before moving to Imperial, and with input from FIMRIB, from cardiology, including not just MR imaging, but also carotid ultrasound, developed a set of protocols for MR of the brain, heart and body, carotid ultrasound, full body DEXA, and a repeat of the whole of the baseline assessment, including biological sample collection. And participants are then going out, the older participants are going out with a 2-week cardiac monitor on to detect in particular AF and other arrhythmias. So we're about halfway through this. We had to pause when the pandemic started, with over 50,000 participants scanned in 4 dedicated imaging centres, one at our coordinating centre for UK Biobank in Stockport near Manchester, in Newcastle, in Reading, and in Bristol. And these data too are being made readily available. Just an example of collaboration within Oxford that I think has been very powerful between geneticists, statisticians, and the brain imagers. This paper that was published in 2018 in Nature, looking at associations, genetic associations with various imaging measures here, genetic associations with hyperintensities, indicative of what was thought to be microbleeds in a particular part of the brain. And these associations of the 4 hits, if you like, on the Manhattan plot with different parts within that part of the brain. And all 4 of these were genes related to ion transport and storage, which led Paul Matthews to raise a hypothesis as to whether these really were microbleeds or whether they were indicative of ion deposition in these parts of the brain that were then associated with cognitive impairment and perhaps opening up opportunities for novel therapeutic targets. Building on the success of the initial imaging project, we went to potential funders for the idea of repeat imaging at large scale. The Dementia Platform UK project, led by John Gallagher, had secured funding from the Medical Research Council for a relatively small scale repeat imaging in UK Biobank, and we had done about 3,000 of those repeat imaging assessments before we had to pause the UK Biobank imaging because of the COVID pandemic. But that showed that about 60 to 70% of participants who had gone through the initial imaging visit were willing, despite that initial imaging visit being about 4 to 5 hours and involving up to 100 miles of travel to get to the imaging assessment site, about 60 to 70% were willing to come back and do it again. And so on the basis of that experience, we were able to secure funding from the Medical Research Council, Chan Zuckerberg, and Calico to start a repeat imaging study with the aim of doing so in 60,000 participants, with the addition of optical coherence tomography to the other imaging, and additional samples for hematology and single-cell RNA sequencing, which is the study I mentioned that Nicole Saranza at the Sanger is piloting at the moment. This repeat imaging study with not only repeat imaging, but repeating the assessments of cognitive function, will allow very large-scale assessment of changes in a wide range of imaging phenotypes over a 2 to 7 year period, an average of about 4 years. So one can look at genetic and lifestyle determinants of changes in imaging-derived measures, which will be very powerful, but also identification of early markers related to subsequent disease risk. So are there brain structural changes that precede cognitive decline? And change is likely to be much more sensitive, more powerful determinant of than a single measure at a single assessment visit. So that raised for us the idea, well, if we're going to look at changes in the people who've been imaged, what about looking at changes in the whole of the cohort? And I think this is where we're very keen to get input from the dementia community. So could we get 300,000 to 400,000 of the 450,000 surviving participants to come back for a relatively streamlined assessment visit, perhaps of the order of a couple of hours? Could we maximize the response rates by having them travel very short distances, perhaps by having about 40 to 50 assessment centers covering the 22 areas where we recruited, and most people have not moved? Could we include additional assessments as well as additional kinds of biological samples, and particularly do very detailed cognitive function assessment and look at behavior relevant to early detection of dementia, other measures of aging such as gait analysis, stand tests, and various aspects of frailty. And again, as with the repeat images, changes in risk factors are likely to be more strongly associated with development of disease. Changes in functional health outcomes would be more strongly associated with previous exposures. So this could very substantially increase the power of UK Biobank to study conditions such as dementia. The third dimension I just want to come on to is around duration. So we now have about 15 years of follow-up in UK Biobank, and it is that combination of size, depth, and duration, which I think is particularly unique. There are new studies being set up. Our Future Health study of 5 million people in the UK is being planned, about 1 million people in the All of Us cohort in the US, but they're only in the initial recruitment or even planning stages. So they are always going to be some 15 years or more behind. So at the moment, I think we need to kind of look to see how do we make UK Biobank as valuable as possible for the kinds of research you want to do. And at this point, I just want to use the example of the Million Women study, another extraordinary cohort set up in Oxford by Valerie Burrell, Gillian Reeves in population health, where a national cohort of UK women aged 50 to 64 were recruited when they came for breast cancer screening. So 1.3 million women were recruited between 1996 and 2001, 1 in 4 of all eligible women. And they've been resurveyed through questionnaire every 3 to 5 years. Blood has now been collected prospectively from about 50,000, and genotyping and exome sequencing data of those individuals will be available soon. They're all linked in to electronic health records for cancer, morbidity, and deaths. Now, although this study was initially set up to look at the determinants of cancer in women, it is, I think, now becoming increasingly valuable for other kinds of research, and in particular for dementia. So among these 1.3 million women, so far, more than 35,000 incident dementia cases have been detected through linkage to health record systems. And what they've found is that the symptoms of dementia cause many of the individuals to change their behavior long before diagnosis, which distorts short-term associations and may have led to many misinterpretations of the direction of associations, with reverse causality causing some serious problems in interpretation. So this demonstrates this really rather beautifully. what it is looking at is the relative risk of dementia onset among women who are inactive versus active at baseline. So within the first few years of follow-up, inactive women have a higher risk of dementia than active ones. But with longer follow-up, that disappears. And as indicated here, what this is showing is this decreasing bias, decreasing reverse causality, whereby the onset of dementia or cognitive decline is influencing activity rather than the other way around. And so in order to really study the determinants of dementia, it's necessary, at least in this case, to exclude the first 10 years and then look at associations after 10 years. And by doing that, by excluding the first 10 years and then looking at the associations, what one gets is really very nice and specific associations. So although these diagnoses are determined through linkage to health record systems, what you can see are specific associations that are different for Alzheimer's versus vascular dementia in a way that you would anticipate. So here we look at current versus never smoker, association with vascular dementia, really very little and probably merely residual confounding for Alzheimer's or misdiagnosis. For hypertension, likewise. For diabetes, likewise. And for activity, no association for either Alzheimer's or vascular dementia. So I think sitting within our research community here in Oxford, we have a number of cohorts, and I would say the Million Women study is a particularly valuable one because it has this very long-term follow-up. It has very large numbers of cases, 75,000 dementia cases already, 33,000 diagnosed as Alzheimer's, 19,000 as vascular dementia. And so I think the ability to measure biomarkers in those individuals for whom there is a blood sample provides an initial opportunity to look to see whether we can understand the genetics of dementia at scale. So I think watch this space, but also do consider collaborating with Jill Reeves, Sarah Flood and others in the Department of Population Health on this project. Moving back to UK Biobank again, we are behind the Million Women studies, both in terms of scale and follow-up, but we have linkage to death data, to cancer data, to hospitalization data, and increasingly now to primary care data. If we look at the trajectory for Alzheimer's disease, then by 2027, there will be some 17,000 cases, and by 2032, some 37,000 cases of Alzheimer's in UK Biobank from health record linkage. There are opportunities to link to other kinds of data beyond death, cancer, HES data, hospitalization, discharge data, and primary care data to get more information about all sorts of different kinds of health outcomes. We're looking at various pilot studies to enhance the information that we can get from record linkage, and smartphone apps is one area where one might be thinking about ways in which we could assess cognitive decline. And we also use web-based questionnaires repeatedly over time to reassess changes in health outcomes. So in particular, repeating our cognitive function web questionnaire, for whom about 200,000 people complete that every few years, will allow us to get information about changes in cognitive function. The scale of the numbers of individuals who've developed particular conditions is huge, obviously. And so the existing methods of trying to characterize health outcomes using the full medical records in a subset of participants is slow, labor intensive, not really scalable. There are ongoing efforts to map codes from different sources to a common coding classification to enable broad scale pheno-wide analyses. But we think there are real opportunities for machine learning and AI approaches to combine data across diverse health records, to combine it with other kinds of information that we can obtain about the participants to get much more detailed and specific assessments of health outcomes. And again, I think this is an opportunity where the Oxford community could help. People such as Chris Holmes are helping us to develop data challenges, and we're talking with various groups around data challenges, looking to get more specific classification of dementia conditions in UK Biobank. Finally, access. For those of you not familiar with UK Biobank, it is available to all researchers, both commercial and academic, for all kinds of health-related research. And the data are really very readily available. The biological samples are depletable, and so we are much more careful about controlling those, but are trying to get the samples turned into whole cohort data so that they're then readily shareable. We're seeing increasing use of the data. Initially, it was pretty much predominantly in the UK. When the genetic data became available, there was an increase in the US and in mainland Europe. In the last couple of years, we're starting to see increases from China, as well as from other high-income countries, but very little so far from low and middle-income countries. Increasing numbers of publications based on UK Biobank. In 2021 alone, some 1,700 papers using UK Biobank data, and increasing numbers of citations to those data. And to finish off, I just want to mention that as part of the whole genome sequencing project, we were provided with funding to develop a cloud-based platform that has been built for us by DNA Nexus, such that researchers can go to the data rather than have the data downloaded to them, particularly as the scale of the data has increased. And that platform is now in existence, and more tools are being added to that platform to make it more usable by an increasingly wide range of researchers. As part of the development of that, AWS, the Amazon Web Services, are providing $500,000 per year of free computing for early career and low and middle income country researchers, and we're looking to enhance that use. So to conclusion, we have this combination in UK Biobank of size, depth, duration, and accessibility. I think with your help, there are many opportunities to enhance the resource for dementia research, and I would very much welcome your thoughts now or at any time to advise us as to what we should do to make it more useful for dementia research. Thank you very much.
Thank you, Rory, for this comprehensive overview. And I must say that I never realized before coming to Oxford that this was possible, that you can assemble a consortium to study also, for instance, metabolomics or other omics data in UK Biobank. There are various questions. The first one, I think you almost already answered, which is a question of Veronica Cabrera. She asked, is there a way to tackle patients in the pre-symptomatic phase with cognitive function? And I think you alluded to that is available. But it's being improved, and she wants to know if there are any patient self-reporting dementia diagnosis, and what about biomarkers in an early stage?
Yeah, I mean, be very interested to know what things should be measured. I mean, I was sitting in the Dementia Summit that was held a few weeks ago, and as I listened to that, I was thinking, well, what do I know whether any of the markers that are interested dementia researchers are on the Olink platform? So during the meeting, I sent a message to Olink. They told me they only had total tau. I contacted Henk Zekkerberg and Paul Matthews and said, is that any good? They said, no, you need the specific. I went back, and then they said, well, we're actually thinking about adding the specific p-taus on. So I think that we need to actually tell the people who are developing these platforms what it is this community wants on it. I think what we're asking from UK Biobank is if we could get funding, and I think we have a very good chance of doing so, of getting funding to get everybody back. What is it we should do? What samples should we collect? What measurements should we do? What assessments should we do? And so we're already trying to bring together a small initial working group to brainstorm that and then go out to the wider community. But I think there is a momentum to build on the success of the last 5 years, enhancing the resource, to enhance it further for particular purposes. I think the other thing is that the web questionnaires we do, which are obviously active, they require someone to do something. And whether with UK Biobank participants, who I think are very altruistic and very engaged in the project and trust the project, there may well be the potential to get them to put something onto their phones, their smartphones, that would allow them to assess them passively, particularly as their habits change.
Yeah. I think there's more markers in Oling that are relevant. For instance, GFAP, what was discussed in CSF this morning by Charlotte Teunissen, and NFL, which is a marker of neurodegeneration, is also in the platform of Oling. But what is missing is, of course, p-tau as the big biomarker. There's another question from Claire McKay, and she asked about the inception of the Biobank. How was it perceived when Biobank started? Was it supposed to be of interest or ridiculous or outrageous? And how long did it take to convince funders to commit to something on this scale?
Well, it's very difficult to find out why the MRC and Wellcome Trust did decide to fund UK Biobank. And I've heard from insiders that people at Wellcome Trust said UK Biobank will be funded over my dead body. I know that Richard Peto and I, during the 1990s, put in applications to the Wellcome Trust to set up such a study. I think people like Nick Day in Cambridge did as well. And that was formally rejected. But then suddenly this decision got made, and it really is impossible to find out who made it and why they made it. There was certainly in the early part of this century a lot of opposition. There was opposition from GeneWatch, who thought it was bad to be studying genetics in population. There was opposition from MPs, in particular the chair of the Health Select Committee, and also from pretty prominent geneticists. And one of the things I had to do was explain that, in a sense, it wasn't a genetic project. It was a standard epidemiological project where one could look at the determinants of many different diseases, because the opposition from geneticists was case control would be much more cost effective.
That's what I recall, that argument. Why don't we do another Wellcome Trust GWAS-like model?
Yeah. But I think, and that's true if you're only interested in genetics, but as I said at the beginning, I think it was is the combination of genes, environment, lifestyle that a resource like this, provided you have the scale, provided you have the resources, and the MRC and the Wellcome Trust really have been extraordinary in being willing to put in substantial funding with a very long time span, then as a resource, these resources become increasingly valuable, as is the case with the Million Women Study.
All right, I'm a bit mindful of the time. Vanessa Raymond says that, well, UKB has been invaluable to the deep and frequent phenotyping study, enabling the participants to be included through that line, and that is, I think, fantastic. John Todd argues that we all have to submit Bombard O-link for our proteins. We will do so, John, don't worry. So we'll ask for what is possible. And Masoud Hussain asked how many comprehensive how comprehensive is the linkage of UK Biobank to the NHS data?
So death, cancer, and hospitalization is complete. Primary care has been a problem. We did get access to all the primary care data for COVID-related research under the emergency powers that the Secretary of State was able to implement. And the discussions that are now going on is whether those emergency powers can be extended to other serious health conditions. And I think that that is getting some traction. But primary care data would, in dementia, as the Million Study investigators have shown, help us to pick up dementia cases more rapidly than we get through HERS, although they have shown that the HERS data does pick it up as well. They've done a very nice piece of work on that. I think going beyond the health record linkage, using web questionnaires, using repeat assessment, would enhance it enormously.
Yeah, there's one last question, because we have to be mindful of the break, is that John Davis asks, are there equivalent of your UK Biobank? The answer is, of course, no, not yet, but there are a few.
Well, I mean, just before UK Biobank was set up, Zhengming Chen in population health set up the Chinese Kadoorie Biobank. That I think is not as deep, and it struggled because of the data access issues with the Chinese government. I think if that can be resolved, and it may be that the research analysis platforms will help in that way, in that people can go to the data rather than data leaving the country, then I think there's a potential for a lot more assays there. The study I think that I would draw attention to is the Million Veterans study that Mike Gaziano set up within the US Veterans Program, because that does have a very heterogeneous population, both in terms of socioeconomic class, ethnicity, mainly male, but very comprehensive linkage. And Gemma Hopewell in the Big Data Institute has been working closely with Mike Gaziano, and I think is another, if you like, person with whom others within the Oxford community should collaborate, because there are very few people who've managed to get access to those data. So I think we're incredibly well placed to work on this.
And you have, of course, also FinRisk as an alternative, but it's all more limited and it doesn't have that deep phenotyping, right? It's all linkage more.
Yeah.
All right, I think we have to close this session.